Here we are back for a second look at Chapter 13 on viruses from our microbiology textbook. And we left off last time talking about the animal virus sort of life cycle. How does it go about, oh, there I go, go about replicating itself using the machinery of a host cell. This is just a cartoon version to get us caught back up to where we were. So we have a virus, and it has a protein on the surface that can bind to a receptor on the host cell, and we see that the virus is now going to enter into the host cell. We kind of skipped some possible cartoons that could have showed it entering the host cell because we've already jumped ahead to being inside and uncoding, removing the capsid proteins from around the viral genome. And now we have the viral genome, in this case a DNA genome, and it's going to transcribe some messenger RNA. It can be used to produce some viral proteins in the cytoplasm, all these little balls, and also replicate the genome. So we have a lot of new copies of the genome, a bunch of virus proteins, capsid proteins, enzymes, and so forth. Then we have the maturation phase where we're going to assemble a bunch of virus particles, typically in the nucleus for the DNA virus, and then finally release. Those virus particles are going to bud off of the surface and take with it part of the cell membrane along with some virus proteins that were inserted into the membrane. Perhaps if it's an envelope virus. If it was a lytic virus, it would just burst through the cell membrane and out would come all the virus particles. What if it was an RNA virus? Things are just this tiny bit different. You should just keep a logical eye on what would have to happen for an RNA virus to mature, and if you guessed based on that, you'd probably be right. In this case, we're talking about a plus-strand RNA-containing virus. So we haven't talked that much about plus and minus strands. If you remember, the DNA is double-stranded, and the two different strands are anti-parallel. They're running in opposite directions, and the plus strand contains codons that could theoretically encode for the transfer RNAs and therefore the amino acids that make up a peptide chain. So in this case, we have a plus strand RNA, so it can act as a messenger RNA. And so here we have the virus coming into the cell, being uncoded, and then we see some virus proteins are immediately being synthesized using that genome because it's a plus strand. It's a single-stranded virus, plus strand. And then we're going to use an RNA-dependent RNA polymerase, which of course normally animal cells do not have anything like that, and produce some minus strands, and then that'll allow us to produce a bunch of new plus strands to use for packaging up viruses. So we can't make any copies of the plus strand until we make a minus strand, right? Because RNA would have to be synthesized from a complementary strand. And so we finally wind up with a bunch of virus proteins and virus genomes. The maturation phase are going to be packaged up, and then release of the brand new plus strand, single strand RNA viruses. Okay, what about a minus strand RNA containing virus? I should have mentioned coronaviridae are such viruses, single-stranded RNA viruses that have the plus strand as their genome, and we'll see those in just a minute in a table. Now we're talking about minus strand RNA single-stranded viruses, and so what's different about that? Well, first of all, the minus strand can't be used to encode can't encode any new virus proteins. These are actually the capsid proteins and so forth from uncoding. I pointed to those erroneously here. These are the capsid proteins, but over here we have synthesis, biosynthesis. We have new proteins being formed. So coming back to this step, we know that these can't be new proteins because this is a minus strand RNA, and it can't be used to translate into protein. So the first thing we got to do is make some plus strands, and then from some plus strands we can make more minus strands, which are the genome. And from the plus strands we can synthesize a bunch of proteins, and now we have capsid proteins, enzymes, and so forth, like genomes. Maturation phase, assemble those things all together into virus particles and let them go. The biggest difference is that we can't get around to synthesizing any protein until we make some plus strands from that minus strand genome. And of course, we can use those plus strands to make more copies of the minus strand genome. This is just a summary chart showing all the possibilities. Um, Here's a picture that we didn't really talk about, a double-stranded RNA virus. The minus-strand, single-strand RNA virus, an example is rhabdoviridae, and we'll take a look at those in a table in just a second. We'll have a quick example of those. Reoviruses have double-stranded RNA genomes, so it's not that different. We're just going to have to use the plus strands to copy and make some minus strands to copy and make some plus strands, and then we can synthesize protein from the plus strands and then assemble some virus particles and release. Now we come to retroviruses. Retroviruses are a little different. Retroviruses are single-strand RNA viruses. So happens they have multiple copies, but that's not really critical. They have a unique enzyme called a reverse transcriptase. So we know that animal cells have RNA polymerases that can transcribe RNA from DNA. They have DNA polymerases that can make copies of DNA from DNA. But this enzyme is unique. It can make DNA from RNA. That's the reverse of what happens in animal cells. So we have this RNA genome, and the reverse transcriptase enzyme will make a DNA copy, and ultimately a double-stranded DNA copy of the viral genome, which will be incorporated using an enzyme from the virus right into the genome of the host. Excuse me. So now we have a provirus, just like we did when we saw the lysogenic cycle in bacteria with bacteriophage. We have a provirus that has the genome of the virus in there, and that cell can live indefinitely with that genome in there, or it can go ahead and transcribe messenger RNA from that genome and produce new viral proteins and new viral genomes, and then package up and mature some virus particles and release them from the cell. In this case, by budding. These are enveloped viruses. But just so you know, there's this reverse transcriptase step happening in here, and we're going to use that in the laboratory because we use reverse transcriptase when we purify all of the messenger RNA from a cell or a population of cells. We can then reverse transcribe all that messenger RNA into cDNA, which, remember, there's no introns, and then we can amplify any gene we want from that original cell using PCR amplification. So that's a pretty majorly commonly done but extremely powerful method in genetic technology and molecular biology even today. All right, so it's just a comparison of bacteriophages and animal viruses. The big difference really is just that bacteriophages inject their DNA into the host cell. Animal viruses have to enter the host cell and be just having a mental block. Uncapped. Freed. Free me. All right, here's an interesting topic. Certain viruses can cause cancer. One example is a retrovirus. Viruses that can incorporate their genome into the host cell genome, into the host cell chromosome, can cause cancers, because if the integration site happens to disrupt the function of an important gene that controls cell cycle, cell replication, cell division, we can wind up with a cell that's no longer showing controlled division of cells. So integration of a retrovirus, once its double-stranded DNA is produced, it can activate what's called a proto-oncogene. Proto-oncogenes are just normal genes in a cell, in the genome of a species, that helps control the cell division, the cell cycle it's called. Are the cells growing or not? And so these are growing activator proteins that are normally turned on at the appropriate time to let the cell divide when there's a growth factor present, a peptide outside the cell saying, yes, time to grow. Well, that's the normal function, but if our virus integrates right in a part of the control region of that proto-oncogene, so that now it's always turned on instead of only turned on when there's a growth factor, now we have a tumor cell, and that's the starting of a cancer potentially. So that's a way that integration can lead to cancer. Proto-oncogenes being converted to oncogenes, which are genes that make cells transform, turn them into tumor cells. Some viruses carry their own oncogenes, genes that can turn on the cell cycle. They kind of mimic proto-oncogenes, so they turn on the cell cycle and make the cells grow ad infinitum. And so the simian virus 40 virus has what's called the large T antigen, and that will trigger unabated cell growth and tumorigenesis, transforming cells. Another thing that can happen is tumor suppressor gene disruption. Normal cells carry genes for what are called tumor suppressors, and they're genes that become activated when the cell grows really wildly out of control. The cells have surveillance mechanisms to say, wow, that's not a normal amount of growth. That's wrong. We should just check out. The cell should just kill itself because it knows, I use that anthropomorphism kind of loosely. The cell has this mechanism to prevent completely abnormal growth, and it will activate cell death. That's a tumor suppressor gene. And P53, for example, is one you've probably heard of in human cells that triggers cell death when the cells either have too much DNA damage, so they're probably going to have a lot of mutations and not be reliable, or they're growing wildly. Something has turned on a growth pathway in excess of what it should be. They'll just kill themselves. Well, if we eliminate that tumor suppressor gene and have a cell that's growing abnormally, now we've got a recipe for a tumor cell. So those are a couple ways that the DNA can be integrated into the genome of the host cell and cause cancer. There's something else I was going to say about that. Anyway, if it comes to me, I'll stop and we'll take another look at it. So, oncogenic DNA viruses, a few are listed here, and oncogenic RNA viruses, including retroviruses that have that reverse transcriptase enzyme that can convert their RNA genome into DNA and integrate into the host. So, but again, not only our retroviruses are not the only viruses that can cause cancer, as you can see listed on the left. All right. The difference between a acute infection, a latent infection, and a persistent infection. An acute infection is what happens when a virus binds to the surface of a host cell, goes through all those life cycle stages we talked about. We have biosynthesis, we have maturation of a whole bunch of virus particles released. We have this huge number of virus particles present in the milieu, blood, or wherever. Then eventually, virus production will slow down if there's not another batch of host cells to infect. But sometimes there's a persistent infection, a slow, steady infection of cells and production of viruses that leads to ongoing worsening disease over a longer period of time. That's called a persistent infection. Latent infection, some viruses initially infect some cells, a large number of virus particles is produced, and then there's no further virion production for a long period of time, sometimes even years. HIV virus may be latent for 10 years in someone's body, and then suddenly the provirus will then reemerge or reactivate within the genome of the host cell and start producing virions again, and we have a latent infection. Human herpes viruses do similar things. They suddenly become reactivated and start producing a lot of virions again and symptoms of disease. Well, we haven't talked about symptoms yet, but so again, latency is an idea that some viruses can be dormant or latent for a period of time. They're not showing any sign of being present. Human herpes viruses and HIV are great examples of retroviruses in general. They can sit with their provirus incorporated in the genome of the host cell. Human herpes viruses do it differently. Instead, they go into neurons. They go into neurons and they stop growing for someone. They lurk or reside in the cell body of some neurons and just sit. So in your body, if you're a child who has someone who had chickenpox as a child, you have some neurons in your body that contain fully functional human herpes viruses, and they're just sitting there. And Why, we don't know, but at some point those viruses may become reactivated when you're 65, 70, 75 years old, and come right back out down the axon to those neurons into the epidermis and cause lesions in the epidermis, painful sores, and that's called latency. Shingles is that disease when you have the HHV3 virus that causes chickenpox reactivating, causing shingles. Persistent viral infections. An example, subacute sclerosing panencephalitis. Measles virus, which we'll see in a table in just a moment, causes a childhood disease. Hopefully not anymore because we have vaccines. It's a disease that potentially has this terrible outcome. Most people that get measles virus get sick for a week or so, and then they get better, and that's all there is to that. However, some children that get measles, or adults that get measles, wind up with this fatal um, uh, long-term persistent viral infection. And so it's a very terrible thing indeed. So it's so terrible when someone loses their child, and a reasonably high frequency of it, fairly rare, but still too many to just ignore. All babies in this country, as much as possible, are vaccinated against measles for that reason, so we don't have this persistent viral infection taking place. Here's some some important viruses that you should probably at least know something about. Adenoviruses cause common colds. So, respiratory infections, common cold. One of the several viruses that very often causes common colds. It's a non-enveloped DNA virus, and adenoviruses are used in the laboratory research all the time to introduce genes into cells to study the function of those genes. So you can take an engineered adenovirus genome in the form of a specialized plasmid, insert a gene of interest into it, and as well as a selection marker, and then grow up a whole bunch of those plasmids, and then transfect them into a packaging cell and produce a whole bunch of adenovirus particles, and then use those to infect the target cell of interest and make them express or knock down a gene and study its function. So anyway, the adenoviruses. Papillomaviruses, HPVs, human papillomaviruses, are very important because they cause genital warts, which is a sexually transmitted infection, but they also can cause cervical cancer. So that's something significant. So HPV 16, HPV 18, and a few other strains can cause cervical cancer, and so hence the advent a few years ago of a HPV vaccine to prevent those viruses from doing that. Smallpox is a terrible, terrible disease caused by vaccinia virus that had such a high mortality rate. It was just unbelievable. It wiped out large populations of people throughout the world during periodic epidemics, and so terrible that a great effort was made to control smallpox, control vaccinia virus, and it was the only disease-causing virus that has been entirely eradicated in the entire world. There are no free-living smallpox viruses anywhere in the world. There's only 2 places in the world where there are still complete competent virions in freezer storage, in the United States and in Russia, in research labs where they had been studied. And the question always rages, the debate rages about whether those last bits of vaccinia, smallpox-producing vaccinia viruses should be destroyed so that there's no chance of somebody getting a wild hair and introducing smallpox back into the world, or should we maintain them in case there's a mutation in a virus that leads to a disease that could be helped by studying the smallpox virus some more. Very fascinating stuff. Herpes simplex, human herpes virus 1 and 2 are very important because cold sores, they have latency, so cold sores can pop up periodically throughout the lifespan because those human herpes viruses can become reactivated and cause lesions. Again, they live in, the sensory neurons of your skin. They also cause lesions on the genitalia. So HHV1 and 2 can be spread by sexual activity, and so we can have these lesions either on the around the oral cavity or in the face, the eyes, even, or in the genitals. Human herpes virus 3 causes the childhood disease chickenpox. And again, we do vaccinate for chickenpox. If you grew up in the United States, most likely you've been vaccinated for chickenpox. It shows latency, and later in life it may reactivate and cause shingles, a very awful disease from what I've heard. HHV4 is called Epstein-Barr virus. Epstein-Barr virus is extremely common. I forget what the frequency is. I think it's estimated as something like 30 or 40% of the population has been infected with Epstein-Barr virus and would show signs of the DNA if they were tested, but there's no effect of it. But sometimes Epstein-Barr virus can cause secondary effects such as lymphomas or leukemia or chronic fatigue syndrome. So Epstein-Barr virus. It also causes mononucleosis. I should have mentioned that. That's a disease that teenagers often get. Not often, but sometimes. Most people that get Epstein-Barr virus infections have no lasting effects at all, but some people, and don't even get very sick, but some people get very, very sick from it, may be sick for a month or more. And that's called mononucleosis. And again, people that have had a severe case of mononucleosis are more likely to develop sequela or later complications associated with that experience. Not everyone, let me just scare you. It's pretty rare. All right. There's another human herpes virus, cytomegalovirus, HHV5, I believe it is, that's much like HHV4 in that a large fraction of the population has been exposed to cytomegalovirus. It causes a harmless, mild disease, mild flu-like disease, or asymptomatic disease. And again, many of you probably have even had it. The reason it's even worth bringing up is that people that develop immunocompromise, such as people with human immunodeficiency virus and AIDS, the reactivation of the virus can be especially vigorous and cause all kinds of problems. Human herpes virus 8 causes a cancer called Kaposi sarcoma. It's a type of skin cancer caused by this virus, but only in people that have immunocompromise. And in fact, Kaposi sarcoma does exist in nature independently, but it's so rare that the incidence of several people getting Kaposi sarcoma in a small geographic region at the same time is one of the key findings that initiated a full-out search for a viral cause in the case of human immunodeficiency virus before they even knew what this was, back in 1980, I don't know, 1 or 2, and they began to catch on that there was a virus behind the HIV syndrome, AIDS syndrome. Hepatinovirus, hepatitis B is an important virus. It causes liver infections, liver inflammation, and left to itself, it will cause a chronic liver inflammation, which can lead to either liver destruction, cirrhosis of the liver, or liver cancer. So this chronic inflammation of the liver is not a good thing, and hepatitis B is so prevalent and so bad that luckily there's a good vaccine for it. And if you work in a healthcare setting, by law in the United States, you have to be offered a hepatitis B vaccine because you are very likely to be exposed to it in the workplace, and it's best to be vaccinated against it so you don't have to worry about getting hepatitis B. Polio virus, I'm not going to go into detail right now, except to say that polio virus was once upon a time, before vaccines were developed, an extremely common disease that was a digestive tract transmitted disease that in a small fraction of the population, about 1% of people that got polio would wind up with paralysis, damage to motor neurons that control skeletal muscles, and damage paralysis. This was a very bad disease, and it has been essentially reduced to almost nothing by very effective vaccine campaigns. Coxsackie virus causes a fairly severe sickness. Common cold viruses, rhinoviruses, are another type of virus that cause colds along with adenoviruses. Hepatitis A virus causes an acute liver infection. Hepatitis A virus will resolve on its own and doesn't last a long time like hepatitis B. All right. German measles is a childhood disease that is pretty relatively mild disease involving a rash and sickness in children, but there's enough cases of more severe damage by German measles that there's a vaccine against that. Measles, mumps, and rubella. That's a three-part MMR vaccine that all children get in this country, if at all possible, to prevent rubella infections. Measles infections, come to that in a moment. Yellow fever and dengue fever are very, very severe diseases that are endemic and are common in certain parts of the world. If you travel outside the country, one of the key things you should do, no matter where you're going, you should consult with a healthcare provider and ask them, what, if any, vaccines should I have, or remedies should I know about, or preventive measures should I take as I travel to these new parts of the world that have different types of diseases that are common, so that I don't get sick. If you get yellow fever or dengue fever, you'll be sorry. I'm sorry to say. Don't do it. You got to be careful. You got to protect yourself. All right. Hepatitis C virus. Hepatitis C causes a chronic inflammation of the liver, which up until recently was a death sentence. Only about 15 years ago, a successful, effective chemotherapy was developed for hepatitis C, so that no longer do people die basically of liver failure that get hepatitis C. So that's a pretty major breakthrough in medical technology. It's pretty expensive, but it's better than the alternative. Coronavirus, I don't need to say too much more about coronaviruses. They cause a respiratory infection, and SARS-CoV-2 is the name of the virus that causes COVID-19, the most recent pandemic, ongoing pandemic of SARS. Lissa virus is a very terrible virus that causes rabies. You should know about that. Rabies is a virus that hitches a ride on neurons, motor neurons of muscle cells that travels along the axons of motor neurons into the central nervous system, and then starts damaging and destroying the central nervous system. And it's a fatal disease unless it can be eradicated immediately after exposure. Filoviruses cause Ebola. And if you haven't heard of Ebola, you should do a Google search on that. You probably have. A few epidemics of Ebola virus have broken out in the world since this thing first appeared, I think in the 1980s for the very, very first time. It caused severe hemorrhagic disease with an incredibly high mortality rate. Well over 50% mortality rate of Ebola. Marburg is an almost identical virus, identical helical virus, causes hemorrhagic disease. Extremely contagious and extremely deadly diseases. And in Africa, probably about, 5 or 6 years ago, there was a pretty bad epidemic that took a long time to get under control. A lot of healthcare workers died. A lot of people died. I think there's still a base level of Ebola virus infection going on there now in Western Africa. All right. Mumps is a childhood disease that you get vaccinate for. Measles, mumps, rubella vaccine again. Measles, morbillivirus measles has a fairly high incidence of that. Well, not high, but enough so we don't like it. That long-term disease, that panencephalitis disease that I was talking about earlier. So we vaccinate against rubiola. So measles, mumps, and rubella vaccine is used to prevent this disease from spreading in the population, provide herd immunity against measles. All right, influenza viruses. Influenza type A is the most common, but it causes a well-known disease that's not very pleasant, and it spreads around the world, and it takes a toll on elderly and very, very young humans, and so we vaccinate as much as possible against it every year. Try to look around the world and see which strains of influenza, right? This thing mutates so fast that every year new strains are coming around the world, and plus the different parts of the influenza genome just keep reshuffling with other strains of the virus when they get common infection of a host. brand new strains are produced that can have new host ranges and new degrees of disease causing or virulence. So every year, an effort is made to produce a vaccine that will be useful against the particular strains that are circulating that particular year. So that's the deal with the flu vaccine. Worth getting it, even if it's not as high efficacy as say the COVID-19 vaccine, which in itself isn't quite as efficacious as we might like, but still it's better than being guaranteed almost to get influenza whenever the cycle comes through the community. Lentiviruses, as we said, are the type of virus that cause human immunodeficiency virus that cause AIDS and certain cancers. All right, so that's our story about viruses. We have one more thing to talk about, I think, which is really remarkable. One of the most bizarre things, I think, in all of microbiology and immunology is the idea of prions. What is a prion? It's a proteinaceous infectious particle. Let me just clarify. It's a protein. A protein that on its own, with nothing else with it, can go from one person and infect another person, get inside their body, and cause disease all by itself. Right? How complex is a virus? Not very. And yet, how simple is this? This is a single protein. Remarkable. So, if you ingest prion proteins from someone with disease or some animal with certain diseases, then you can wind up getting a prion disease. And typically the prion diseases that we're going to mention are horrible, fatal, nervous system destroying diseases. So, right, if you get an organ transplant, better hope that they did their homework and you don't get a prion infested organ, or else you're going to have prion disease. Surgical instruments can be infected with prions. Very, very hard to get rid of because it's just a protein. And there's resistant to almost anything you can do to it except add some denaturing or some enzymes that can actually break down the protein into amino acids and so forth. So not that easy to destroy prions. Pretty terrible stuff. So Creutzfeldt-Jakob disease comes from, well, there's genetic predispositions, but there's also bovine spongiform encephalopathy, which when people consume the nervous system remnants from a cow that had bovine spongiform encephalopathy, they may develop Creutzfeldt-Jakob disease, a degenerative disease of the brain. So what's going on with it? Let me go to a picture that says everything that this slide says, only it's easier to follow in a picture, I think. So prions are misfolded versions of proteins that are normally present in the body. So here's a cell that makes this particular protein. We're going to call it PRP, and it's the normal form with this superscript C, and it's just part of the normal structural function of the cell. And so these PRPCs are on the surface of the cell. Along comes a misfolded protein. It's called SC for scrapie because one of the early discoveries of prion diseases was scrapie, which is a sheep prion disease. Imagine crazy sheep being turned loose. So it's the scrapie protein. It's the abnormal form of the protein, and if you become exposed to it, it will bind to the normal proteins and cause them to misfold. And then those misfolded proteins, as they accumulate, every time a misfolded one touches the normal one becomes misfolded, and so they accumulate and they get endocytosed into the cell, but the cell can't degrade them because they're abnormally shaped and so forth, and they accumulate in the cell in vesicles and eventually kill the cell. It's not unusual. There's quite a few diseases called, quote, storage diseases, in which cells accumulate vesicles full of stuff they can't deal with. Eventually the cell dies. So that's the deal with how prion, the single protein coming into your body, can ultimately lead to the demise of these host cells that normally produce this PRP protein. Remarkable. What cells are we talking about? We're talking about neurons. And so neurons in your brain are destroyed, and physical holes are forming in your brain, and there's loss of degeneration of brain function, loss of brain tissue, and so forth. So that is just craziest stuff in the world. On the next section, you can take a look through this last section of the PowerPoint. It just goes through pretty much the same viruses that I just talked to you about in that table that I showed you earlier. It's kind of nice because it maybe gives one or two extra details, but just take a look through that table and it's another chance to refamiliarize yourself with a bunch of those important viruses that you should know something about from coming out of this chapter. So that concludes our discussion of chapter 13. We'll go on and talk about epidemiology in chapter 14.